talked about your CHOP and your GAP, and it downregulates your protein synthesis. That leaves you with your naked mRNAs and RNA binding proteins, which eventually leads to your formulation of your stress granules. Now, the stress granules, as we said before, is kind of what leads to your inability to go from your short-term to your long-term memory. So therefore, their next question is they wanted to see what can prevent these stress granules and how these stress granules cannot be formed by inhibiting and looking at the effects of your alpha-2A phosphorylation.
Okay, so they looked at two different ways in which stress granules can be formed to see if it's alpha-to-alpha phosphorylation dependent or independent. So first, if you prevent the phosphorylation of alpha-to-alpha, as expected, stress granules would not form. There wouldn't be an accumulation of mRNA. But also, if you inhibit L4-alpha, which is actually used to go into the stress granules, so it's one of the mRNA present, you would never produce stress granules as well. So they look to see if you look at the PAD-A or the HIP, if RIB's able to minimize stress granules formed through those, or if it's only able to inhibit stress granules formed through alpha phosphorylation.
And so then they looked at the formation of your stress granules, and in order to visualize, they used L3A or L3 alpha and our DAPI. So our DAPI is just used to visualize the cells, and our L3A or L3 alpha is present in stress granules. And so, as shown here, it might be okay. So we had a little bit of a hard time visualizing this, so it might be hard to see, especially up here on the screen. But what they tested, the stress granules are these the little green. It's not like the big overall green. It's these tiny green dots are the stress granules. And so you have your stressful conditions, and then you have in presence of your RIB. And so this shows that there is a decrease in the tiny green dots with the presence of RIB under your L2 alpha phosphorylation. And then this is just the quantitative data for that, showing that your presence of RIB has a significant decrease in your stress granule formation under the L2 alpha phosphorylation, and what that phosphorylation causes.
And so what was ours? ARS?
That is a it's a stress condition that is caused by your L2 alpha phosphorylation. So they're testing to see if the stress granule formation is dependent on your L2 alpha phosphorylation. Yes?
Why didn't they use the TM or the TG like they were before? Why did they start using ours?
Because the TM and TG, I believe, are just like overall ER stressors. They're not specific to the L2 alpha phosphorylation, because they're looking specifically, does stress granules form from your L2 alpha phosphorylation compared to your L4 alpha inhibition. So I think they wanted to like specifically see. Does that make sense? Because the TM or the TG.
Well, they're using the TG.
I mean the TN, I don't think is as specific as the ARS. Is that?
Yeah.
Yeah.
Change that. It wasn't.
And I know TG is stronger inducing, stress inducing, so maybe that might have something to do with it.
TG
TG
is
is a lot
strong.
stronger, so I don't know if it's strong enough to get the stress granule response they're looking for.
No, that's
It
this one.
wasn't the same.
Yeah.
Could you turn on the lights, please?
Yeah.
It was the same set of.
To the top.
Okay. I'll get it, guys.
Yeah, you want the TG because you want to be able to see the granules being able to be formed. And so TG, as they said, is a very strong inducer, so you can see induction. TM is not a strong enough inducer of ER stress to form granules. It'll still shut down sort of protein production, but it won't form the granules. Now the R's, that I have to look up because I'm not. Anybody else know what what R's does?
It's a use inducer of alpha-2-alpha phosphorylation.
Okay,
So that's.
so it's another strong inducer.
Okay, so they're testing like in this case is specific, and you'll see like the other one they used to test specifically the alpha inhibition of the next one.
Okay. Any other questions? Does that make sense? Okay.
Okay. So the next, as we said, they wanted to test. So this is the quantitative for what we just looked at.
Is that the last fluorescent one that you have?
The next one. No,
Next
we have one more.
Okay,
Yes.
okay.
Okay, and so then the next test they did, we'll show you the graph first and then the fluorescence. The next one, they wanted to see the PAT A in your hip. Those are L4 alpha inhibitors, and so they wanted to see if RID had an effect on those those phosphorylations, and if those phosphorylations cause stress granules. So as shown here, so they used just how they use ARS. So the R's was a inducer for L2 alpha phosphorylation. Now your PAT A is an inducer for your L4 alpha inhibition. So those are the two things that they're comparing here. And shown here is you can still see the presence of those tiny green dots and those stress granules in the presence of RID. Therefore, it was concluded that your RID did not have an effect on your L4 alpha inhibition, and it only affected the stress granules that were formed from your L2 alpha. So the next thing we want to talk about is, why are we talking about stress granules? Why do we care? And what does this have to do with memory?
Okay, so memory consolidation is your ability to go from short-term memory to long-term memory. And to accomplish this, first you have to have an increased synapse strength to be able to fire your signals, and also you need to be able to have your neurons repeatedly fire and be able to fire in like similar sequences. But whenever you're under stressful situations, you develop stress granules, so this accumulation of mRNA, your protein's not being synthesized. So as Aubrey touched on earlier in the presentation, your actual channels that between each neuron, you require proteins. So if you don't have enough proteins, your signal's not being able to efficiently be fired from neuron to neuron. So this prevents long-term memory from being developed. So overall, big picture, this research is really important because under stressful conditions, we're unable to retain information, which is obviously really inconvenient, especially with finals coming up. So if they're able to figure out how to still allow long-term memory to occur although your cells are experiencing stress, it'd be very beneficial.
So now that they made these conclusions with these different tests, they wanted to see how it's going to affect memory in mice. So the heterozygous mice they presented, they said that they have enhanced memory. However, with the L2 alpha phosphorylation in presence of PKR, which is an L2 alpha kinase, that the brain cells, it impairs their memory. So they wanted to test this, and they actually did, and we hope you guys find it interesting too. They did a water maze along with some other tests. So to give you a little bit of a background, we have a video to show you of the Morris water maze that they used. Should I even try the volume? No? Okay.
So initially, they have an elevated platform in the water maze, and they allow the mice to swim around and recognize this platform.
And there's only about an inch above the water that the platform sticks out. And so the mouse has to find the platform, get up on the platform, and realize that when it's on the platform, it'll be rescued from the water maze. So it has to swim around, find it, and then get up on it to be rescued. And then, as they showed, the water after they train the mice. Then they put the mice back in, but they don't have the platform sticking out of the water. So the mice have to swim around and remember where the platform was and still be able to get on the platform.
And they also dye the water, so the mice aren't able to see where the platform would be.
So when they train the mice, they train them, they can see the platform even from under or over the water. And then when they test them, it makes it harder for them to see, and then it's not sticking out. So in their test that we're talking about, they use this water maze to test the memory of mice. And so what they did was they gave the mice this rib, and then they trained them through the water maze. And then they did this whole procedure, and they tested to see how long it took the mice that had rib and the mice that didn't have the presence of rib, and the different times it took for them to find the platform. Does anyone understand kind of how they did that? Okay.
We just found it really interesting, so we thought we showed you.
These are rats, right?
Yeah.
That's a big mouse, that's a.
Yeah.
Okay, so what they did, I said they injected the mice with this rib, and they trained them through the water maze, and then they compared the effects of the mice treated with rib and mice that weren't. And they measured the latency, or the time it took for the mice to recognize the platform. What's really interesting to note is that, so they did these over a period of, they trained the mice, and then they tested over the 5 days. Now you can see that the mice that were normal and not treated with the rib, at first, the first 2 days, there's not too big of a difference in the time it took them to recognize the platform. Because, as we showed, that this affects long-term memory. It affects the ability for your short-term to be turned into long-term memories. So therefore, you can see that over right when they're trained, the short-term memory isn't, as nearly affected as the long-term memory is.
Okay, and then another test that was very interesting that completed was a tone shock. So they placed mice into a small area, and they gave them like a high-pitched tone. And then right after the tone, they shocked the mice in the feet, and they got them used to that stimulation. Well, then they took the ISR, IB, or the rib mice, and compared them to just regular mice to see if, when placed back into that same situation and they hear the tone, if they would freeze, if they would remember the shock that was associated with it. So, as this data shows, similar to the water maze, the rib are actually,
So this is your short-term memory, and this is your long-term memory.
yeah, so they were a lot more effective in recognizing the tone shock and freezing for longer periods of time. And also in the short-term memory, it wasn't as much of a difference. So they were both able to remember the shock initially, but as time went on and you were to place the rib mice into the same situation, they were going to remember the shock and remember the pain associated with it.
So we definitely think, as we were looking at the different studies that they did with the mice, how important and how great of results they got with the mice studies, because this just really shows the effect that this rib can have on long-term memory. And so going forward with next research, Dr. Miller actually heard this guy talk at the biology conference, and he was talking about how his next step is to give the mice like blunt head trauma and to see if this rib can really reverse the effects and reverse the memory loss and to gain back memory. So it's really interesting, we thought, especially from the mouse studies, to see the different effects it has on memory. So overall, in conclusion, this rib is very beneficial going forward in studying what effects stress can have on your memory consolidation and making short-term memory go to long-term memory, and the importance it can have on having you remember even though you're stressed.
Any questions?
Cool.
Yeah.
So yeah, they are basically